Thanks very much, Steve, for the off-the-cuff introduction, and thanks for the invitation to be part of today's proceedings. It really is an honour to be part of it. Extraordinary company I find myself in, and I'm a little overwhelmed, I have to say, to be following people of such incredible calibre. I'm going to talk today about our work on cell death pathways and a new class of cancer drugs called the BH3 mimetics and the impact they have on a lineage, a blood cell lineage that's dear to the heart of many people in this room, including Hatem, and that's the megakaryocyte lineage, which is responsible for producing platelets. So once platelets are produced by megakaryocytes in the bone marrow, they are shed into the circulation. Platelets are essentially anuclear fragments of megakaryocyte cytoplasm. Once they're shed into the circulation, they undergo one of two fates. A section of those platelets are consumed in clot formation, in haemostasis, but we make a lot more platelets than we actually need, at least at steady state. And so for the vast majority of platelets, their fate is to be consumed by the reticuloendothelial system in the liver and the spleen. So this was first noted by William Duke in 1910, who was performing some of the early blood transfusions and published a paper in the Journal of American Medical Association, a seminal piece of work where he noted that upon transfusion, unlike red cells which lived for weeks and sometimes months in his patients, platelets lived for only a few days. And in the 1950s, when cancer chemotherapy really took off, people became really quite interested in the issue of platelet lifespan and pinpointed the fact that in humans, circulating platelet lifespan is approximately 10 days. And later on, it was established that in mice, it's 5 days. And the mechanisms that underpin this finite existence in the circulation have been the subject of speculation ever since. So our insight into this process came from a very simple genetic screen conducted in mice for mutations that impact on platelet production and platelet function. I won't go into the details, suffice to say that this is really just a chemical mutagenesis screen, essentially accelerated evolution, which allows you to produce a cohort of mutagenized offspring and allows you to screen them for phenotypes of interest. And in our case, that was mice with thrombocytopenia. We've isolated a number of pedigrees and subsequently identified a number of genes important for platelet production and function. And the two that are relevant to today were given the names PLT16 and PLT20. And they were two mice that presented with thrombocytopenia that turned out to be heritable. And both mutations upon mapping mapped to the same region of the mouse genome, mouse chromosome 2. This summarises the mapping data. This is really almost obsolete now in the age of exome sequencing, but this was a few years ago. Essentially what it tells you is that we identified two independent mutations in the gene BCLX, which encodes this protein here, BCL-XL. So we had two mutations in BCL-XL, both of which caused thrombocytopenia. And really, the critical question at this point was why. So BCL-XL hadn't been linked to platelet production before, and so I need to introduce, at this point, the BCL2 family of pro-survival proteins, of which BCL-XL is a member. There are 5 BCL2 family pro-survival proteins: BCL2, BCL-XL, BCLW, MCL1, and A1. And their job in cells is to restrain the activity of these two proteins here in orange, Bak and Baks. And Bak and Baks are the executioners of the intrinsic or mitochondrial apoptosis pathway. So when this system is in equilibrium, a healthy cell survives. When a cell is subjected to a stress such as DNA damage, the BCL2 family, the pro-survival proteins are inactivated, allowing Bak and Baks to be unleashed. They cause mitochondrial damage, efflux of cytochrome C into the cytoplasm, and that then leads to caspase activation and apoptosis, or programmed cell death. So this is a process which has been studied really in extraordinary detail over the last 25 years, particularly at the Hall Institute. And it's been very well established, this basic paradigm of pro-survival proteins restrain the activity of Bak and Baks in nucleated cells. So it occurred to us eventually that perhaps BCL-XL might be regulating the survival of an anucleated cell in the platelet. And so we measured platelet lifespan in vivo. This is very simply done in mice via a pulse chase experiment using biotin labelling. And when you do so, these are platelet survival curves in mice in vivo. And you can see that, as I said before, platelet lifespan is approximately 5 days. When you look at mice that carry one mutant allele of BCLX, platelet lifespan is reduced to 3.5 days. And when you look in mice that are homozygous for the mutation in BCLX, platelet lifespan is reduced to only 24 hours. And we went on to show that these effects are intrinsic to the platelet, they're not an effect on the host. So as you incrementally reduce the function of BCLX, you incrementally reduce the lifespan of platelets in the circulation. Now at this point, we were able to complement the genetic work with some pharmacology and ask what effect the pharmacological inhibition of BCLXL might have on platelets in vivo. And we were able to do so in collaboration with Hall colleagues David Wang and Andrew Roberts, who were doing a lot of work at the time with a compound called ABT737, which was developed by Abbott Laboratories specifically to target the BCL2 family pro-survival proteins BCLXL and BCL2. So the rationale behind this is to inhibit pro-survival proteins in cancer cells, which have typically evolved the ability to evade normal apoptotic signalling. And at the time we were doing this work, a relative of ABT737 called ABT263, now known as navitoclax, was about to enter clinical trials. So we were interested to know what effect this drug would have on platelets. We injected mice with ABT737, and what we found is that in contrast to mice treated with vehicle, mice treated with ABT737 suffered a very severe and rapid onset thrombocytopenia, and we went on to show that this is because the drug is directly toxic to the platelets themselves. So the genetics and the pharmacology matched up quite nicely. At this point, it was pretty clear to us that BCLXL is very important in the regulation of platelet survival, and that immediately suggested that its job was to restrain one or the other, or both, BAC and BACs. So we turned to the BAC and BACs knockout mice and measured platelet counts. These are wild-type platelet counts. In the BACs knockout mice, platelet counts were normal, but in the BAC knockout mice, they were thrombocytotic, and that immediately suggested that BAC is important in the regulation of platelet apoptosis, and by extension in the regulation of platelet lifespan. If you look at platelet lifespan in the BAC knockout mice, shown here in yellow, relative to the wild type, shown in blue, you can see that there's an almost doubling of platelet lifespan in vivo, and this is the first genetic manipulation which has been shown to extend the circulating lifespan of platelets. So at this point, we turned back to ABT737. We injected BAC knockout mice in blue, or BAC knockout BACs heterozygous mice in orange with ABT737. And you can see that in contrast to the wild types, the BAC knockout and the BACs knockout mice are refractory to the effects of ABT737. So again, the pharmacology and the genetics match up. BCLXL is required for survival, and BAC and BACs are responsible for platelet apoptosis. So to put these things together to explain how platelet lifespan is regulated, we've come up with the molecular clock model of platelet lifespan. Obviously, we believe that BCLXL is required for platelet survival, and its job is to restrain the activity of BAC and to a lesser extent BACs. Now, our model is that a megakaryocyte endows a set amount of BCLXL, BAC and BACs on an individual platelet. Because they can't synthesize new protein, they are dependent on protein degradation mechanisms, and we know that BCLXL degrades more rapidly than BAC and BACs. So the model is that as the platelet circulates, the pro-survival protein degrades, and at 5 days in mice, 10 days in humans, there's no longer sufficient pro-survival activity to restrain BAC and BACs. BAC and BACs become activated, they damage the mitochondria and cause apoptosis and clearance from the circulation. So it's a very simple model to explain how the body can limit the lifespan of an anucleate cell, and a cell type that's potentially very dangerous, and a cell type that the body produces more than 100 billion of every day. So that was a few years ago, and a number of implications and questions arose from that initial study, and the first and most obvious was that BCLX inhibition in patients was likely to cause thrombocytopenia. As I've mentioned, ABT263, now known as navitoclax, was going into clinical trials, and those trials were able to be conducted in the knowledge that thrombocytopenia was a likely side effect, and that this was an on-target effect of the drug, and that indeed turned out to be the case. The major dose-limiting toxicity of navitoclax has proven to be thrombocytopenia. The drug itself has shown a lot of promise, and with lead-in dosing to circumvent some of the severe thrombocytopenia, it's now moved into phase 2 clinical trials, but there's no doubt that its development has been slowed, and that Abbott's perhaps enthusiasm for the drug has been tempered by that. And these data really encouraged Abbott, in collaboration with colleagues at Weihai and Genentech, to develop a second generation BH3 mimetic known as ABT199, which has recently entered clinical trials and is showing really very exciting efficacy in chronic lymphocytic leukemia. ABT199 only targets BCL2, and it was developed to specifically avoid hitting BCL-XL. And Genentech and Abbott were using thrombocytopenia in dogs as their surrogate marker for BCL-XL specificity in developing this drug. So the knowledge that BCL-XL is essential for platelet survival was really useful in the clinical development, not just of navitoclax, but in the development itself of ABT199, which is recently published and showing, as I said, very, very exciting activity in CLL. I think it's important to note at this point that this is just the tip of the iceberg with the BH3 mimetics. There are a number of companies with a raft of different BH3 mimetic drugs at various stages of development, and they have differing profiles of activity, targeting different members of the pro-survival proteins in different permutations and combinations. So this is a class of drugs which is only just beginning to see its potential realised. And I think it does highlight the importance of understanding the role of the pro-survival proteins in the megakaryocyte lineage, given that thrombocytopenia is such a common occurrence in cancer patients. The next most obvious question really was then regarding megakaryocytes, the platelet precursor cell. It seemed likely that megakaryocytes themselves were probably dependent on BCL2 family pro-survival proteins to stay alive. And so to that end, over the last few years, we've been testing that hypothesis, deleting pro-survival proteins specifically in megakaryocytes using a transgene that expresses CRE specifically in the earliest committed megakaryocyte precursor. So we started with BCL-XL because of its essential role in platelets, and we found that when you knock out BCL-XL completely in megakaryocytes, platelet counts are essentially zero, which makes sense and fits with the idea that XL is essential for platelet survival. But after a lot of work, we realised that it wasn't just platelets that require BCL-XL for survival, but megakaryocytes themselves, just at the point at which they make platelets, require BCL-XL. So this is a wild-type megakaryocyte in culture undergoing the final stages of platelet production. This is a process called proplatelet formation, and you can see that the cytoplasm, essentially the entire cytoplasm, forms these very elegant, very sophisticated cytoskeletal pseudopodia called proplatelets. And in vivo, it's these filaments that project into the circulation and from which platelets themselves bud. So that's what a wild-type megakaryocyte does in culture, and this is what a BCL-X knockout megakaryocyte does. And you can see that just at the point where they're starting to begin to make proplatelets, they undergo apoptotic cell death. So just as platelet production is about to occur, megakaryocytes become totally dependent on BCL-XL. But they grow and they develop normally up to that point, which obviously suggests that there are other pro-survival proteins at play here. We know that BCL2 is expressed in megakaryocytes, and we know that MCL1 is expressed in megakaryocytes. So we have deleted MCL1, and we find that alone MCL1 is not required for megakaryocyte development nor platelet lifespan. But we can use ABT737 then on top of this to inactivate BCL2, BCL-XL and BCLW in megakaryocytes. So if you take a mouse which has no MCL1 in its megakaryocytes and you treat it with ABT737, what you find within 2 hours of treatment is that the megakaryocytes basically melt. So this is a wild-type megakaryocyte sitting there smiling at us, and these ones are undergoing apoptotic cell death, and you can see these magnificent pycnotic nuclei as the megakaryocytes condense and contract and undergo apoptotic cell death. And you come back 4 hours later, there are no megakaryocytes in these mice. So we think this is telling us that MCL1 and BCL-XL together are responsible for the growth and development of megakaryocytes. Without them, megakaryocytes can't survive. So now turning to another question which has been occupying our mind for some time now, and this is work that's been conducted in collaboration with Sean and Hatem and a host of others here at the ACBD. We're interested in the idea that by blocking apoptosis, we might be able to extend the viability of blood bank platelets. Blood bank platelets have a short lifespan, just as platelets have a short lifespan in vivo, and apoptosis has been implicated in the platelet storage lesion, the complex degeneration that platelets undergo in storage. So we thought if we could block apoptosis, we might be able to extend their viability, but that obviously raised the question of what that might do to a platelet in terms of its function, and whether it would be actually useful upon transfusion, or worse, would it actually be dangerous. So for the last few years, we've been trying to get at this question of what Bak and Baks might do in platelets. Why? Because if you take an activated platelet and you look at many of its behaviours, to a non-platelet person, they look an awful lot like an apoptotic cell. A platelet treated with an agonist undergoes cytoplasmic condensation, mitochondrial depolarisation, microvesiculation, caspase activation, and phosphatidylserine exposure. And to someone who knows nothing about platelets, that sounds like apoptosis. So we thought it was worth exploring what Bak and Baks might be doing in platelet function. And we started with phosphatidylserine exposure. And as many of the people here will realise, phosphatidylserine is employed by activated platelets to facilitate the assembly of the tenase complex, which drives the production of fibrin. And if you don't have PS on your platelets, you suffer from a bleeding syndrome called Scott syndrome. But in apoptosis, phosphatidylserine is exposed by the cells to recruit phagocytes to facilitate engulfment. So you have a scenario whereby a platelet presumably exposes PS to drive coagulation. Does it also expose PS to drive apoptosis and cell death? And is there a link between the two? So this is work done with Sean and Simone in particular. If you take wild-type platelets and you treat them with ABT737 to induce apoptosis, you get a really nice PS response. So indeed, an apoptotic platelet, just like a wild-type nucleated cell, when it dies, exposes phosphatidylserine. And if you start to delete Bak and Baks, this is a double knockout platelet population here, you ablate that PS response, which is not surprising. Apoptotic platelets don't expose phosphatidylserine. On the other hand, if you treat the same genotypic classes of platelets with agonists, and this is convulsin and thrombin, the PS response is absolutely untouched. So this was really the first evidence that platelets have evolved two very distinct mechanisms by which they can expose phosphatidylserine. So just to summarise roughly where we're at, this is a very mitochondrial-centric model of the platelet, of course, but we believe that agonists through cyclophilin D drive the activation of calpains, and that facilitates phosphatidylserine exposure, which is important for coagulation. On the other hand, apoptotic signals lead to the activation of Bak and Baks, cytochrome C release, and caspase-dependent PS exposure, which may be involved in platelet clearance, or that's an open question. The question we've been asking more recently with Sean is whether this is involved in coagulation. Is there any crosstalk between these two pathways? So I'm going to leave you with just a little bit of unpublished data. If you look at hemostasis in these mice, these are the Bak-Baks double knockout mice here, where Bak and Baks have been deleted in megakaryocytes. They have a modest but reproducible bleeding defect, suggesting that platelet function is impaired in these animals. And if you look at thrombosis formation in the carotid artery model, this is an electrolytic injury model. In contrast to the wild-type mice shown here in black, the Bak-Baks mice take a lot longer to occlude, and those thrombi are unstable. So this is intriguing data. It does suggest that there is a role for apoptosis in the hemostatic and thrombotic function of platelets. We don't know why. One obvious explanation might be that in an activated platelet which exposes phosphatidylserine, in a subset there might be a tipping over into apoptosis, a triggering of apoptosis, which may be a mechanism by which platelets can then amplify the coagulation cascade by putting more phosphatidylserine on the cell surface. It's just a model at this point, and I think we've got a long way to go to understanding what Bak and Baks might be doing in terms of platelet function. So just to summarise, I think there are many important roles for the apoptotic pathway in the megakaryocyte lineage. To begin with, megakaryocytes absolutely depend on MCL1 and BCL-XL to grow and survive the process of platelet production. And if you interfere with these pro-survival mechanisms, for instance with chemotherapy or BH3 mimetic drugs, you induce apoptosis and death of megakaryocytes. Platelets, once born, are absolutely dependent on BCL-XL for survival. And they depend on XL to restrain the activity of primarily Bak, because Bak is the end point of platelet lifespan in vivo. Bak mediates the death of platelets in the circulation. And I think we still have a long way to go to understand whether there's actually a functional role for this apoptotic pathway once platelets become activated and are in the process of forming a thrombus. Finish quickly by acknowledging the many people who have been involved in this work. I won't go through them all, but I would like in particular to mention Emma Josephson, a postdoc in my lab, who's now a lab head in her own right in the Cancer and Haematology Division at Weihai, who's done an absolute mountain of work on this project. And my colleagues David Wang, Andrew Robertson, Kile Mason, who've been collaborators all along the way with this project and really have been fantastic supporters and contributors. And of course, this work has involved a lot of people at various times in various ways from the ACBD. And I guess in particular, Sean and Hatem have been have been very generous and have been real supporters, and I think this has been a great example of a north and south of the river collaboration, and we look forward to a lot more of it in the future. And I'll stop there and take any questions. Thank you.